Our well-being is dependent upon the coordinated work of all molecular mechanisms in the body. Many biological processes are mediated by the interaction of special molecules, ligands, and receptors. A disease state results from disruption of these interactions, while their normalization is a key to successful treatment. Antibodies can be used to achieve such a goal. Antibodies are complex proteins that can specifically bind to a wide range of molecules. The development of antibody-based drugs is one of the most promising areas of the modern pharmaceutical industry. Generally, therapeutic antibodies are used to suppress the activity of biomolecules in the organism, preventing the binding of a ligand to its receptor. Recently, scientists have discovered that special technological processing can change properties of antibodies. What are the sources of antibodies used in the drug production? Antibodies to the human ligands are obtained from rabbits of a special breed, grown in sterile conditions and free from viruses and bacteria. The ligand administration results in the synthesis of antibodies used in the further drug production. For safety reasons, the final biomaterial is subjected to sterilizing filtration and precipitation process. The next step is to separate antibodies from other components using affinity chromatography. Antibodies are collected from a special column after binding to a specific ligand. Finally, antibodies undergo quality control in accordance with international and national standards. What are the basic principles of the technological processing of antibodies? The antibody processing technology is based on the latest findings relating to the aqueous solution properties. Scientists have discovered that mechanical or electromagnetic treatments have an effect on the properties of water systems. This occurs due to formation of various nanoscale structures in the liquid. What is antibody processing technology? Drug manufacturing technology is based on the most recent advances in biology and physics. Purified antibodies are subjected to gradual processing using a microreactor. Inside its microchannels, the antibody concentration is gradually decreased, which is accompanied by a special physical treatment. As a result of the technological processing of antibody solutions, nanoscale structures are formed. The final solutions may contain original antibodies due to the effects of flotation. This technology represents an advanced approach to new drug development. The final product is used to manufacture solutions and tablets. What is the process of production of the finished dosage form? To obtain tablets, technologically processed antibodies are first applied to excipients. Then they are tableted using a standard method. Final product quality control is carried out using modern analytical methods. What is the principle of drug action? When reaching its targets, the drug changes the ligand or receptor shape, which affects the interaction between the participants of biochemical reactions in the body. The drug components, as well as the technological parameters of their production, are selected to enhance or weaken these interactions, depending on the type of dysfunction. Drugs have a specific therapeutic effect, which means that technologically processed antibodies modify only specific molecules without affecting others. Drugs have passed a full cycle of non-clinical and clinical studies. This makes it possible to manufacture drugs with high therapeutic potential.